Thank you so very much for that overly kind introduction. I hope you can see my slides. I would have liked to be there in person. I really regret not being able to join you in person. There's so much more one can do in the real world, but this is a next best solution, and I'm glad to be here and to tell you a little bit about my background, my work in brain networks, and there are actually two lectures. One today will focus on structural networks. That's on anatomical networks, wiring diagrams, the kinds of things we can do with graph theory to understand the architecture and the organization of brain anatomy for the most part. And then tomorrow, I will pivot over to functional networks, brain in action, as it were, dynamics, the ever-changing configurations of functional links in the human brain particularly. And I look forward to tomorrow because I will also talk a little about new work that part of it is unpublished that we're very excited about here and have spent the last couple of years working on that deal with dynamics very explicitly. My talk today will be somewhat expository and didactic, as it were, probably going over some material that many of you are already familiar with. I'm sure there are people in the audience who have much more profound knowledge of graph theory and networks than I do. But as the introduction stated, my interest is in applying these concepts and tools and approaches to the brain. And to do that properly, we also have to understand the brain and how it is structured and how it functions and how we measure it. So a lot of what I talk about today and tomorrow will be viewed through the perspective of a practicing neuroscientist. And rather than blindly applying, high-level mathematical and computational concepts to the brain, I'd like to motivate the choices that I have made in my past work through viewing it through the lens of understanding how the brain actually works. So today, at first, a very brief introduction to this intersection of network science and neuroscience, which is a thriving field. And I used to have to motivate what I'm talking about by arguing that the brain makes a perfect system for understanding it as a complex system and as a network. Happily, this has receded, and I think it's generally accepted today that the brain is a complex system, and that complex system methodology, models, and tools and methods can be profitably applied to understanding brain structure and function. At a very fundamental level, the brain is a network. It's not just a metaphor, I believe, or an analogy, but it is literally a network of neurons interconnected by synapses, or brain regions interconnected by interareal projections that we can measure and map at different scales of resolution. More about this later, and then tomorrow especially the dichotomy between structural and functional networks, especially functional dynamic interactions. So far from being just a metaphor or a comparison or a facile analogy, the brain is a network. And so network science methodology is very suitable for understanding how the brain is organized. Network science, of course, is a broad topic. There are many other examples of complex systems that you have probably come across, and perhaps many of you are working on. I take networks to be a natural realization and a real world implementation, if you wish, of complex systems such as the brain. Fundamental elements, of course, are nodes and edges. I'll focus mostly on the nodes today. You will see that there's a pivot in our own work going to edges, and I will talk about this a lot tomorrow, not today. And of course, we're all familiar with diagrams such as these that illustrate the various renderings of networks in social and biological systems, and brain networks have burst upon the scene the last couple of decades in a really major way. Many of you are familiar with graph theory, and I just put this slide up here to illustrate some of the very basic concepts and measures and metrics that we take on board in our field of brain networks. You all know what node degree is. I don't need to belabor this point. The clustering coefficient, something about the local neighborhood of a node and its propensity to be interconnected. Network motifs, there's an interesting relationship here to what Sinestra talked about earlier today on simplicial complexes. I think there is a connection between what used to be called motifs in our field and is still called motifs to that high order hyper-edge configuration model. The importance of communication processes in the brain, to which I will speak especially tomorrow, makes the concept of a path and the path length and distance, topological distance on a network, really fundamental. In this particular toy network here, we need 3 steps to go from A to B, and that metric of distance, which is measured not in millimeters but in numbers of steps, is crucial for understanding communication processes. Again, we will turn to that tomorrow. We will turn today, however, to a couple of concepts down below. One very fundamental and often misunderstood, that's what I want to spend some time on it today, and that's modularity or community structure. A decomposition of a larger network, perhaps a very large network, into a smaller set of components that are internally densely connected and more weakly connected between. That concept, simple as it may seem, is actually very difficult to quantify in a data-driven way, and is often misunderstood in neuroscience. So I want to take a little bit of time today to go into some of the methodological details and the recent methodological developments that speak to the importance of the concept in our field. A corollary of modular decomposition is also that it allows us to identify critical elements of the network architecture that are critical for communication between network communities. So this node here illustrated in black, if we lose it, you can imagine if it's attacked by a pathological process or injured in an accident, that node will, the loss of that node will have a disproportionate impact on communication processes in this network, as all connections between these two modules are lost as a result. So I'll come back to the importance of hubs later today and also tomorrow. Finally, the concept of a rich club or a subgraph that consists of high degree, highly connected nodes, which are also internally densely connected with each other. A concept that again comes from graph theory and has been somewhat impactful in the neuroscience space. Why? Because of its potential importance in integrative processes in the brain. More about this later today and tomorrow. I take it that you're familiar with these concepts, but I wanted to just define my terms up front so that we're clear as to what I'm talking about later. Now, network science and network neuroscience, as we now call it, has is undergoing a dramatic expansion. And there's a reason for that, and that is in neuroscience, we have access to more and more refined and more dense recording methodologies and mapping methodologies. We can observe the brain at different levels of scale, from molecules and interactions at the millisecond level all the way to long-term processes that occur in a person or in a population over a period of days and months and even the lifespan. Brain processes play out on all these different levels of time and spatial scale. And we can observe them with a variety of technologies, many of which are fairly recent and have allowed us to map brains and monitor and record from brains with ever greater precision and coverage. For today, I will largely stay in this middle ground, covering recording and mapping methodologies that allow us to map brains at the level of neurons and systems. And tomorrow specifically talk about diffusion MRI and a functional MRI, which allow us to map brain activity at the whole brain level. This is the space in which Connectomics resides, a term that we came up with 15 years ago now, and that has actually expanded dramatically in our field. There's a fundamental distinction in neuroscience between structural and functional networks, and in fact, it runs through my two lectures. Right? Today I'm going to talk mostly about anatomical and structural networks. Tomorrow I'll talk mostly about functional networks. I want to make clear that everyone understands what that distinction is and why it's important. We can obviously observe many different types of brains, all the way from simple, relatively simple creatures such as the nematodes, the elegans, to the human brain and non-human primate, in between rodents and macaque and so forth. And we can do so in two fundamentally different ways. We can map the structure, that is the anatomy, the wiring diagram of a brain, or we can map what it does through time. On the left, structural connectivity is measured typically using some sort of anatomical postulation. If we look at the whole brain, breaking the brain down into regions that are internally fairly homogeneous and externally distinguishable. And we can then define connections or projections between them by measuring tracts, axonal connections between remote neuronal populations. We can do that with a variety of methods, from tracing, histological tracing, and invasive methodology can only be used in model organisms, or non-invasive imaging, and which is largely done in humans. The end result of that is a sparse, sometimes symmetric, sometimes directed matrix of coefficients that defines a graph. And methods from graph theory are readily applied, have now been applied very widely to graphs and networks derived from almost any model organism you can imagine. And most of my talk today will be about what we've learned from doing so, and highlight some of the commonalities and differences across different species. On the right is a very different workflow. We're recording from the brain. We're recording its activity over time. This gives rise to these, voltage or bold signal time traces, wiggly lines as I call them. And we can then apply various tools from time series analysis, dynamic modeling, etc., but also networks to understand what is happening with respect to the statistical dynamic interactions over time in a brain as it is either spontaneously active or undergoes particular task-evoked processes or cognition. The result of that analysis can be, in the case of human neuroimaging, very often still is, as simple as a correlation matrix that is often called in our field a functional network. And again, tomorrow we'll dig into what this is and how to deal with it. I want to say upfront that graph theory and a lot of the methods coming from topology are readily applied, I think, to wiring diagrams and structural networks, shown here on the left. But they are not as readily applied, and in fact, often jarringly inappropriate when used in functional brain networks because of the nature of the edges, the nature of the measurement process itself, and what the edges and nodes represent in a functional network. Graph theory is not a perfect tool. One thing that applies to both worlds, I think, and can be used profitably, and we have used in our own work across domains, is modularity and the understanding of the modular organization of brain networks. That can be applied on both sides, but a lot of graph theory really only applies to the left part of this diagram. This is often neglected in our field, despite the fact that I always say this in my public lectures. And I really want to underscore it here. We have to keep in mind in neuroscience where our data comes from. It is not a good idea to take high-level concepts from math and physics and apply them blindly to brain data. We have to understand where data comes from, what it represents, what the measurement process is, the uncertainties involved, and what actually is the neurobiological process we are intending to interrogate. The construct on the left primarily is the construct that we refer to as the connectome, the structural network, the wiring diagram, and I'll explain in a minute what exactly we mean by that. How do we construct brain networks? Once again, it's important to know, in fact, 90% of the time, and I would say in our lab, is spent with understanding and working on the details of how we actually construct brain networks from primary brain data, from recordings of anatomical observations. In the case of a structural network such as the one shown here, extremely simple and kind of network we had available to us about 20 years ago now, what's involved is measuring through experimental measurements, histology, which brain regions in the macaque brain, colored here in sort of rainbow color scheme, are connected to what other brain regions. And that can be done with so-called tract tracing experiments, invasive injection of viral tracers into brains, which are then histologically reconstructed. The animal, unfortunately, has to be sacrificed in the process. This is not something that can be done in humans. It can only be done in non-human species. Important, when we then construct a network from these data, there are important simplifications that occur. And these are important to keep in mind. Quite often, we represent a brain region as a single node. That's already a massive simplification. A brain region may contain hundreds of thousands, perhaps millions of individual neurons, and these are simplified into a single point. Intra-regional connections are often disregarded in these inter-areal matrices. We are often using binary connection weights as opposed to weighted, although that's changing in the last decade or so. And we usually have very little information about things like conduction delays. Communication in the brain is not instantaneous. It doesn't occur at the speed of light, but at the speed of conduction, of axonal conduction, which is much, much, much slower, on the order of a meter a second or so. There's no distinction often in our data between absent or unknown connections because the measurements themselves are piecemeal and cobbled together from multiple observations. So there are many, many issues that we have to consider when we deal with the kinds of brain networks that we are working with in our field. Very quickly, a few historical notes and broad conceptual points that I'm sure many of you are familiar with. Everyone knows what an adjacency matrix is. Adjacency in a graph simply summarizes which nodes are connected, here indicated by black squares in this directed matrix. This matrix was available to us already about 15 years ago, coming from the macaque visual and somatic motor cortex, showing the interregional connections among different brain areas in the macaque. Reachability signifies whether or not we can find a path of any length between a pair of nodes. In the case of the macaque brain, the brain is fully connected in that sense, forms a connected component, and the distance matrix tells us something about the distance, the topological distance, the number of steps needed to go from A to B, important communication processes. You all know that if we shuffle the order, if we treat a graph as a sort of a lookup table, what's connected to what as a matrix representation, we can shuffle the rows and columns of that matrix without changing any of its topological attributes, really. Here's a random reordering. It is perhaps obvious to you that the ordering on the left is more meaningful. That's because the visual areas are at the top and the smarter motor areas at the bottom of this graph. I want to make a simple point that even something as simple as reshuffling or reordering the nodes and edges in a graph can actually massively illuminate us in terms of its organization. In fact, community detection of modules is nothing else, in my view, as finding a good ordering scheme, and we'll say much more about this later on. We can visualize graphs as matrices, a format that I personally prefer because I see all the data in a sort of top-down fashion, or we can use graph embedding algorithms that have been around for a very long time. Random graphs are probably dear to many of the mathematicians in the audience. In some ways, that's where graph theory got started, and have properties such as a uniform degree distribution. If it's an Adjacency graph, classically constructed here with a single scale of degree, and if we study random graphs as Adjacency did in classic, seminal studies half a century ago now, and we take a large number of nodes and gradually add connections to them, some very surprising, well, to the naive, to the layperson, surprising things can happen, such as phase transition that very quickly results in a connected component in such a graph from a totally unconnected state. These phase transitions are, of course, important for brain dynamics, and I'll come back to talk more about them tomorrow. In our own field of brain networks, we owe a lot of gratitude to our colleagues in the social sciences. Brain networks were not invented by brain scientists, but really many of the concepts that we still use, that I illustrated earlier, really come from the social sciences. And I want to show this because it shows us that in social network analysis, a lot of the thinking about networks was driven primarily by data sets, by collection of data in the real world, often rather small by our current today's standards. And much of the thinking of these pioneers in social network analysis focused on not random networks, which was the domain of math and physics in some ways, but on how small, non-random, empirically observed associations between individuals in a social setting can help us understand something about functional outcome, how well that system operates, how well the social structure holds up, how robust it is, how it evolves through time, and so forth. And we've taken that on board, I think, in our own field in brain networks. We are motivated primarily by empirical observation. And whatever our theorizing about networks really is constrained by the kinds of things and phenomena that we are able to observe and uncover in experiment and anatomical and functional observations. And to round out the historical section here, you all know about small worldness. It was an important development in our field also, because it actually burst upon the scene just about 25 years ago. And without going into detail what it is, you all know this, it actually had an immediate connotation with respect to brain structure and function that we articulated here, my colleague Tononi, myself, and Gerald Edelman, some years ago in a review article. Brain architectures, structural and functional networks, in our view, and I still hold that view today. I'm afraid I have not changed over the past quarter century in my fundamental beliefs about what is important about brain structure and function. The brain is a complex system in the sense that it combines in its architecture and in its functioning elements of disorder and regularity. It is a somewhat disordered system, but not totally disordered. So it has that element of criticality, if you wish. It sits right at the boundary between a disordered state and a state which is somewhat ordered and regular. And to understand how it gets there, how it maintains its position in that regime, and how those resulting phenomena, the heterogeneity of spatial temporal states, the non-periodicity, the ongoing flexibility and robustness, how that plays out in actual brain function, that is, I think, what motivates a lot of our work still today. So a few words now about connectomics and some model organisms. And again, I mentioned the connectome already, but just to reiterate, we came up with that term in 2005, and that term did not exist before that date. We published a white paper, a manifesto, if you wish, some years ago, 16 years ago now, that laid out a research program for mapping brain networks in real species, including the human. And the connectome we defined it at the time as a structural description, a map of the complete comprehensive map of connections within a nervous system. That definition still holds today, in my view. There were several motivating goals behind this. We understand still today, I think many people now agree, that to understand a system such as the brain, one must know how it is connected or wired up. There's no hope, I think, for fully interpreting, understanding functional dynamic brain data and their origin if we do not know what the underlying connectome looks like. It is a fundamental requirement. This does not mean that knowing the connectome tells you everything about functional or dynamic brain data. That's that's not, the inverse is not true. But without that, I think we do not have, we're lacking something very important. In that sense, by the way, the connectome has a similar status, I think, as the genome does for biology in general. No, the genome does not explain everything about biology, but we sure do not want to go back to a biology where we don't have genomics or genomic information. I think we're on the same boat right now in neuroscience. We now have connectomes and we can use them for understanding the brain. But of course, it doesn't tell us everything there is to know, far from it. The differences and parallels to the genome are significant. We actually highlighted them in our first article. Just as the genome, which can be sequenced, a bunch of A, T, Cs, and Gs, 3 billion of them in the case of humans. In the case of the brain, we can objectively measure its wiring. We can't technically do so today, certainly not with readily available economic resources, but we will in the future, I'm sure. And so that the connectome has, like the genome, has an objective validity to it. It is real. It is a structure we can measure, and if we measure it twice, if our measurement process is correct, it should come out the same second time. And there are variable dynamics, just as there exist in gene expression. The genome is just a static, in the simplest conceptualization, it's just a static sequence of A, T, Cs, and Gs, just as the connectome is just a connection matrix of which neurons connect to which neurons. When the system is set in motion, of course, different genes are expressed at different times and brain activity fluctuates as well. The connectome is now a word in the English language, so that's good to know. After 15 years, it entered the Oxford English Dictionary as a new word officially a couple of years ago. We can map connectomes across different species. A few of them I've shown you already. C. elegans, a humble organism, a nematode, a worm living in the ground. Other invertebrates such as Drosophila, avian brain, rodents, mammalian species, non-human primates, and of course humans. And I want to spend in this next segment a little bit of time showing you some of the connectome networks that we have in hand now in our field and what they look like, and a few salient results as to their architecture, their structural topology. We've known this connectome, the network of the neuronal network of C. elegans. These are single neuron connections that you're looking at here. We've known this for a very long time, as it was reconstructed in the late 70s and early 80s, painstakingly reconstructed from electron micrographs. And despite the fact that we've known this network for a very long time, there are still new studies coming out all the time investigating various aspects of its topology and function. And this is not a summary of those studies. It can't be because there are too many of them, but just a few salient points coming back to those classic measures of graph theory I mentioned earlier, like degree and modularity, rich club organization, and so forth. At this level of single neuron connectivity, which by the way has been remapped and refined a couple of years ago using more modern reconstruction technology, so this network has been updated. Connection density, quite sparse, only about 4% of all connections that can exist in a network such as this actually do exist. There's high clustering and short path length, the hallmark of small worldness. And we find even at the neuronal level that there are some neurons which have a lot more connections than others, that are more central with respect to their node degree. Subsequent studies have shown modular organization and interconnections among high degree nodes, which are denser than expected by chance. And that is the definition of the so-called rich club phenomenon. Rich clubness is a non-trivial aspect of structural connectivity in that it does not have to exist. In fact, it should not exist, as I'll illustrate later on, because it is actually quite costly with respect to the wiring, the amount of wiring that it absorbs in a given architecture. More about this in just a few minutes. To Drosophila is a model organism about which we know a lot from genetics classically, but then also from behavioral testing and invasive imaging studies, as well as anatomy. In fact, we now possess a neuron by neuron wiring diagram of part of the Drosophila brain that was published, I think, earlier this year, maybe late last year. So this diagram here is much outdated at this point, as it was constructed a few years before that. And it only shows you really brain regions or local processing units, as they're called in this context. Brain regions of the Drosophila brain, how they're connected, directed connection, as you can see here, and a modular organization that's been laid out in a string embedding with a string embedding algorithm. A key point I want to make is that the Drosophila brain, very different, very removed evolutionarily, very far removed from the nematode, has similar topological attributes: small world organization, the existence of high degree hubs, modular organization, and rich clubness. Similar organization seen in the rodent brain. Here, the mouse, which was extensively mapped by the Allen Brain Institute in the middle 2010s and analyzed subsequently in this study here. Again, modular organization, which breaks the mouse brain down into contiguous regions that are densely interconnected internally and have some connotation with known functional subdivisions of the mouse brain organized by sensory modality or behavioral functionality. Rich club organization is present in this brain as well. I have worked over the years quite a lot with my colleague Larry Swanson at USC on the rat brain. One of our earliest studies in that sequence of work has been the rat cerebral cortex, shown here on the right. I'll come back to the rat later on as well, because we're still working on it quite a lot. Just one word about how these matrices are derived. This is a unique methodology because it leverages existing literature, published reports on rat brain connectivity that exist already in libraries, and most of them employing histological track tracing data. And what Larry and co-workers have done is systematically data mine that existing literature and create data sets that are essentially very large Excel spreadsheets. Literally, that's what they are, recording reports about connections being present or absent, as well as their strength and direction between different regions of the rat brain. Those of you who are not familiar with neuroscience, to you, this terminology, these abbreviations that you see here on the rows and columns may make little sense. Perhaps you can recognize the auditory cortex, the visual cortex here, the orbitofrontal cortex, the entorhinal cortex, and the hippocampus, various regions of the amygdala, etc. So these are brain regions that we know exist in the rat brain, and each of the entries in this matrix records already published reports about whether a connection between these regions exists, and if so, what its strength is and directionality. So it's really a very large literature search that gives rise to a matrix like this, and there are literally hundreds, perhaps thousands of data points behind this, this collation of connectivity data. This is something that is done in part by hand, in part manually by people such as Larry and his co-workers who really know a lot about the rat brain. And it can also be done, increasingly is done by data mining and AI systems. These matrices we've constructed, Larry has constructed them across the rat brain, not only the cerebral cortex, but also the basal ganglia, the hypothalamus, the midbrain, and we're now in the hindbrain and rhombic and cerebellum. Interestingly, they have very different topological attributes. Different subdivisions of the rat brain look quite different from each other. Those attributes that we're familiar with, modularity, rich club organization, and highly connected hubs, are largely evident in the cerebral cortex. But I will say, although I don't have a slide on this today, They are not present as much and perhaps even absent entirely in other subdivisions of the rat brain, such as the hypothalamus, the thalamus, or the brainstem or the basal ganglia. Small world is a feature that pops up in many of our conversations and seems to be universal at some level, but it is not universal in the brain. Here's a larger matrix that I shouldn't say in review, it's long published now, that shows the rat cerebral cortex, the bihemispheric version of this matrix. By that I mean we're showing both the left side and the right side of the brain, and the interhemispheric connections are shown in this off-diagonal quadrant right here. So all the connections that you see in this area I'm outlining right now with my cursor are connections linking the left to the right hemisphere. And I want to point this out because those connections are holding the two sides of the brain together. We all know the brain is fairly symmetric in its layout in most species. There are a few exceptions, but certainly in mammals. And very little actually was known up to this point, at the point of the study, about the organization of the interhemispheric connections. They're rarely measured and they are difficult to quantify for various reasons and difficult to trace and track in human imaging studies. In this case, what we see, we actually see this streak of connections running through the main diagonal of the sub part of the matrix. These are connections we call homotopic. They link brain regions on the left and on the right that are actually homologous to each other. The left visual cortex to the right visual cortex, the left motor cortex to the right motor cortex, and so forth. And this analysis here allowed us for the first time to quantify in a mammalian species in the rat the number and strength and distribution of these connections across the entire cerebral cortex. 55 of the 77 possible connections actually existed, and we had a few other interesting observations in this study that show us that connections between the two hemispheres actually are an exact subset of connections that exist within each hemisphere alone. So they, in some ways, they copy what each of the hemispheres doing internally. It gets copied over to the interhemispheric connection. It's very interesting. It's also been shown in mouse since then. Going quickly over to the non-human primate, about which we know a lot, again, because of invasive tract tracing studies largely carried out in the 50s, 60s, 70s, and 80s. Ridge club organization is present in this organism, areas such as prefrontal areas 9 and 46, areas in the orbitofrontal cortex, cingulate, the temporal cortex are interconnected densely and themselves are highly connected to the rest of the brain. That is the hallmark of ridge club organization. Highly connected brain regions that are called ridge, that are also connected amongst each other, forming a club. Again, in my view, a non-trivial aspect of connectivity. Why? Because ridge club regions, which we show here on an inflated macaque monkey surface, ridge club regions are actually widely dispersed across the cortical surface. Despite the fact that they are quite distant from each other in terms of metric distance, they're far apart in terms of, measured in inches and millimeters, they are densely connected amongst each other. There's an important consideration here in anatomical networks, and that has to do with the issue of cost. Connections in terms of wiring do not come for free because they occupy space, they occupy volume, they take up connection delays and time for that reason. They are made from lipids and proteins, so they take up energy to maintain. These connections are not free. They are a little bit like the internet router system, the physical internet in that sense, not the network of hyperlinks, which is largely cost-free, but the network of cables that run under the Atlantic Ocean or across continents to connect different parts of the world with each other. Those cables have physical properties. They have length, they cost money, they cost energy to maintain. So it is in the brain. So the layout of the brain is actually, we believe, very strongly modulated by this cost consideration. I'll come back to that in just a second. In the human brain, many of the properties that you have heard me mention just before, modularity, rich club organization, highly connected hubs, small world organization, broad, perhaps exponential, we're not sure yet, degree distributions, they all exist in the human brain as well. And in fact, this is from our very first study with Patrick Hagman, where these properties were first identified. This was a study we carried out with Patrick not so long ago, really, with 5 individuals. It is completely impossible in our field, current field, to publish a study with 5 individuals. That would be laughed at. But the field didn't exist back then. We kind of got it started, and 5 individuals was just fine, despite the fact that we had 5 individuals only. These features that you see here that we identified in our very first study have held up through numerous replications in other cohorts with much larger samples in different imaging centers in parts of the world. So whatever features you see listed here, they may not mean much. Perhaps they are entirely meaningless. I don't know. I doubt that. But they are very robust, and they can be ascertained and measured again and again and again. A little aside here, I've talked largely about the brain, the whole brain, whether it's the whole nematode brain, the whole Drosophila brain, the whole rat brain, or the whole human brain. But of course, we can also zoom in and record from individual neurons. And while I don't do this work myself, some of my colleagues that I have been fortunate to collaborate with do this work. And I'm increasingly interested in recording data from individual neurons that allow us to tell something more at a greater level of detail about how neurons connect and how they interact and give rise to behavior. Neurons, as you probably know, communicate for the most part with spikes. Spikes are sort of discrete signals, binary, brief, reversals of a membrane potential that we can measure with invasive electrodes. And they have a precise location and time, a precise spike time attached to it. And a lot of the energy in our field of spike analysis goes into understanding the temporal relationships among spikes emitted by different neurons, and what that might tell us about information flow, computation, and so forth. Working here with my colleague at IU, John Beggs, some years ago, we looked at, or John looked at neurons in culture, hippocampal neurons that were put into a dish and recorded from continuously. And he and his colleagues in his lab constructed networks from these spike recordings that were based on these spike trains and temporal relationships between them, using a metric that we call transfer entropy, which is I would call a budget version of causal directed or effective connectivity. Budget, but because it's data-driven model, it's not based on a generative model, but it's based on spike train observations, recordings. Transfer entropy is fairly well established in time series analysis in this context of spike recordings, and when we use it, when John reconstructs these networks, he can derive from the recording data sparse directed graphs that interconnect, in this case, on the order of 100 or 200 neurons that we're looking at in a dish. And interestingly enough, and for me, shockingly, I'll be honest, many of the organizational features that we've seen at large scales in different species, this is actually mouse that we're looking at here, such as hubs and rich club organization, hold true even at the level of single neurons. This, for me, was utterly unexpected. And it's held up over the past several years in multiple other follow-up studies. What this means is that, first of all, there are individual neurons which have many more connections than other neurons, measured in this case with transfer entropy. And those highly connected, rich neurons are also interconnected densely amongst each other, more so than expected by chance. And that's indicated by this curve in red, which shows us the excess of interconnections among rich neurons compared to a null model. And I didn't expect this, to be honest with you, but it does appear that some of these features that we observe at the whole brain level in anatomy carry forward or carry down into the neuronal architecture itself. In ongoing work right now with a collaborator in Germany, Ben Dunn in Göttingen, and my grad student Thomas Varley, we are now looking at recordings from behaving macaque monkeys. Ben has pioneered this approach, has done these recordings years ago. We are reanalyzing those data. This is a top-down view of the monkey brain with the motor cortex M1 shown here in the middle, part of parietal cortex at the right, and part of premotor cortex at the left in the frontal lobe. What Ben has done is inserted electrode arrays, 6 of them, recorded from them simultaneously as the monkey was performing various behavioral tasks. And the fascinating thing here is that we can listen in on parts of the brain at single neuron resolution as an animal actually carries out behaviors. And with Thomas Varley, Thomas has developed very, I would say, very state-of-the-art techniques to dig into the information dynamics, as it's called, the changes in information flow and information theoretic measures that we can extract from spike trains and related this to behavioral state. Work in progress, a draft manuscript on my desk. Hopefully by next year, there'll be more that we can say about it. But it does look that these features that I've illustrated so far, such as modularity and rich club organization, are modulated across time as the monkey undergoes different behaviors. Finally, to round out this section of the talk, I've gone over a number of different species here, from invertebrates to humans. There are some common topological properties. I don't need to mention them again. You'll see them here. Where do they come from? If there's such a universal, similarity between species as remote as invertebrates and humans, what does that tell us? Does it tell us anything about how the brain is organized? You could argue if something is so conserved, it doesn't tell us much. Or you could argue if something is so conserved, tells us a lot. And I don't know, depends a little bit on your personality, which way you look at this. But I believe that one of the major factors driving anatomical connections is the fact that they are physically embedded. The brain is a geometric object. It is not a virtual network. It's a network of real neurons in space wired up with little tubes that are called axons and dendrites that receive information and so forth. It consumes energy in the form of ATP. There are real boundaries here on how the brain is organized. And we believe, and many people really do believe, that when it comes to anatomical networks, taking into account these geometric and energetic factors, aspects of cost, wiring cost, and energy, are critical for understanding how the brain works and how it is laid out. So the interesting idea then, I still am very fond of it. We wrote an article, my colleague Ed Bullmore and I, almost a decade ago now. I still very much subscribe to it. The real brain that we actually observe has that topological in-between status. It is partly a lattice, and that short connections are predominant because they take up space and wiring, and they need to be short. But they're not all short, as it would be the case on the left. Some connections are also made over long distances, sort of like random shortcuts. It is that in between, blending of local and global topological features that make up the complex topology of the human brain, and any brain really that is organized in 3 dimensions. Just as an aside, it probably means that our brains are terribly suboptimal from the point of view of what brains in theory could be capable of if there was no geometric constraint, if there was no ATP that needs to be used every time an action potential is generated. But one of the interesting connotations here is that our brains, because of the severe constraints on cost, are probably occupying a relatively small space of the overall available topological space that brains could be in. And so we should not think of our brains as optimal along any dimension. They're probably very suboptimal, at least with respect to the wiring layout that we physically find. We can discuss that later if you wish. That's an interesting, almost philosophical topic. Okay, last part of today, modularity. And not so much from a highly technical perspective. Modularity in networks is a topic about which one can write, and some have written very long review articles and books even. But from a perspective of what does it tell us about the real brain and how it's organized. And one of the conceptual links I want to make is that modularity really is a feature of complexity, or at least some type of complexity that we happen to find in the natural biological world. This was pointed out a long time ago by Herbert Simon, of all people, who talked about the architecture of complexity in an article later on a book, and defines modularity as near decomposability, the ability to take a complex system and break it up into pieces such that the pieces have internal, some criterion of internal homogeneity applies to the pieces, but they're still connected to each other somewhat. And that hierarchical organization into modules that are near decomposable may well be common to many complex systems and may have favored, in terms of biological systems, their emergence and evolution. And there may be a crucial link also in information processing. Modularity and information processing are strongly linked, I believe, in the case of the brain, which is why I want to spend some time on illuminating that concept from a neurobiological perspective. The way I still think about it, even though this is a diagram from an old review article, a very similar diagram appeared in an article by Carl Friston, actually, same year, is that in our anatomical organization, as we've seen, we have modular communities that are interlinked by hub regions. As the brain becomes active, as it engages in activity, either spontaneously or driven by the environment, there's an ebb and flow, a yin and yang, if you wish, a push and pull between these two extremes of organization. Full segregation, the modules are organized and functioning and internally coherent all by themselves, or the system is fully integrated and is flat with respect to its modularity. We will see tomorrow that indeed that model in some ways that we propose, or I wouldn't call it a model, but this, this conceptual idea that really has gone through my work since the 90s is gathering more and more empirical support. We can now measure and track these episodes of functional segregation and integration through time in the human brain at a very high temporal resolution. I'll show you this is work in progress that I'll show you tomorrow. For now, let's continue to focus on modules, how they are defined, how we may find them, what they tell us about the anatomy. Well, again, in this audience, I don't perhaps need to emphasize this, but finding a good modular partition in a network is very beneficial, if nothing else, because it allows us to visualize the network in a very meaningful way, where internally coherent, densely connected components of the system are now readily visible in either a matrix form or in a energy minimization layout as shown below. The network here on the left is identical to the one in the upper right, except that I have now cleverly applied modular detection and reordered my nodes in a way that actually visualizes that organization in my matrix plot. This is particularly important when we deal with more complex data. This is, of course, the previous diagram was a toy model, but here's an actual recording from a human brain. I've used this example for years now. On the left, a correlation matrix is a functional network. Again, we'll talk more about this tomorrow. But for now, let's just say, it's a functional network coming from a human brain, 200 brain regions. And I'll challenge you and say it's very hard to understand the organization visualizing it on the left. But if you perform modular detection on the same network using an appropriate community detection algorithm, you can extract a series of modules, in this case 4, that make the organization of this network much more apparent. Again, the net in terms of the data itself, the left and right side are totally identical. It's just displayed differently on the right. And modularity has had an enormous impact in neuroscience. It's not sometimes even recognized by practitioners in neuroscience how they are using the concept on a daily basis. And that will be especially apparent in functional networks that we'll talk about tomorrow. Of course, there's a huge field of detecting communities in a data-driven fashion, which is the domain of statisticians, mathematicians, computer scientists. Some very famous people in our field, Mark Newman, Sandro Fortunato, my colleague here at IU, have written about this extensively. And in fact, even today, a new way of defining modules or detecting modules in networks still has a shot at being published in some of the most important journals in the world, because the topic is so important. Clustering and segmentation is one of the major domains of data science, really, and it's applied across the board, whatever data you might imagine, including the brain. In our field, for reasons that have more historical reasons to them, this type of detecting, this approach to detecting communities that we call modularity maximization is one that is quite dominant. We're trying to move away from it, but like the proverbial super tanker, which neuroscience really is, any change in direction takes an enormous amount of time and sometimes energy. So the field at large out there still uses modularity maximization quite extensively, but there are alternatives to this, which I'll show you in just a minute. But just a few words about modularity maximization. Originally proposed, the important modularity score, modularity metric, proposed by Mark Newman and Michelle Gervin some time ago, essentially quantifying in a single quality metric, Q, the proportion of links, the ratio of the proportion of links within modules relative to between modules, and comparing it to a null model, which is built into the cost function itself, assuming random partitioning. And the higher Q, the better the modular partition. That's the idea. At least that was the idea at the time. It turns out this simple formulation has various drawbacks and limitations associated with it. So we're taking a graph and we're coloring its nodes by modularity assignment, by module assignment, and a good quality partition of the graph into modules shown on the right will give us a high value of the Q metric. That's essentially how it works. How to find this good partition, that's the domain of algorithms, and there are various algorithms out there that do that for us, including the domain algorithm, which is widely used in our field. Problems with this approach, because it's a global approach to modularity detection, that is, it looks at the whole network all at once, this particular approach scales quite badly as the number of nodes and edges increases. As the network gets bigger, finding the one single best partition in a network that's really large is actually very, very hard to do. We cannot do this exhaustively. We have to rely on algorithms to do it for us, and they may be subject to the classic, problem of getting stuck in local minima. And so we often have to settle for finding approximate solutions, and often have to do actually many iterations of an algorithm to determine which solutions are good, better, or worse compared to others. So while the definition of modularity may be quite, mathematically, it can be sort of encapsulated very nicely, finding an optimal partition in any real network is actually quite hard, and that's true for brains as well. To make matters worse, there are some problems built into that metric that need to be overcome, or at least addressed. When we're optimizing the Q metric, we may get stuck in solutions that are verifiably false or wrong, as it were. A network such as the one over here, small modules on a string of pearls, the Q metric optimization may get us stuck on finding 5 modules instead of 10. Similar problem exists down below. There are many different solutions. There are many different scales at which modularity may exist in a given real-world network. And those scales, that issue of scale, has been neglected until a few years ago, and is now more recognized in neuroscience. I'll show you some applications in a minute. There's also the problem of what we call degeneracy of the modular landscape, which is here in this case visualized a landscape of solutions, different partitions that have almost equal modularity Q, but actually have different partitioning schemes underneath. So different solutions that the algorithm may find that are actually non-identical, degeneracy in other words. That's a problem in our field as well. We can address them. I'll show you just in a moment how we might do that. Final problem here especially for functional brain networks, is that links can be negative. That we can have negative links, we can have anti-correlations, for example, in a correlation network. And the classic formulation a la Newman and Gervin does not account for negative edges or negative edge weights. We have actually worked out a scheme by which we can take negative edges into account. The issue of modularity degeneracy can be addressed by taking many solutions which have almost equal Q values, Q metrics associated with them, many partitions of a brain network into modules and communities. And what we then do is we take what is called an agreement or a co-classification matrix. That is, we basically average them together, and that gives us a graded a frequency really for each node pair to be associated within the same module over a range of partitions. And this type of heat map or co-classification network is now very commonly used in our work, at least in my own work and the work of my colleagues, although not yet very widely across in our field. So going away from a single hard partition, even though it may be optimal, to an ensemble of partitions, which can then be aggregated together and treated as a co-classification matrix. Just to show you how this works, our simple network here on the left, here's our best optimal partitioning scheme we can come up with using Q maximization. But if we take multiple runs of the same partitioning algorithm and build an agreement matrix, this module up here is very well captured. It's very coherent across multiple runs of the algorithm. But modules down here are somewhat captured, somewhat less precisely on each individual run. And that, I think, reflects their actual organization in the topology. I'm going to skip this and go to the issue of multi-scale modularity really quickly. Multi-scale modularity is something that really was looked at a lot by Santo Fortunato and colleagues. And in fact, we've worked with Santo now applying this to brain networks. Once again, if we have a set of modules on a string of pearls that are quite small and compact, Q maximization may not give us that verifiably and arguably ground truth, but something that is actually not correct. That is an arrangement into 4 modules in this case. And that's because the Q metric has a built-in resolution bias that can be addressed by modifying it and introducing a so-called resolution parameter. This is what Santo has pioneered. Essentially, I think of it as a zoom lens that allows us to zoom in or out on different levels of scale and capture modules that are bigger or smaller, depending on where we look in the architecture. When we do that and we apply this to a rat connection matrix shown here, a structural matrix of the rat end brain, I think is in this case, which is the cerebral cortex and the basal ganglia taken together, a single scale modularity minimization or maximization may give us the arrangement that you see here. But if we apply a multi-scale approach and we ramp our zoom lens from very large to very small settings and then aggregate together the partitions that we find, what we then get is a co-classification matrix. This is a matrix that's naturally bound between 0 and 1 because it quantifies for each node pair the propensity or the frequency with which they are associated in the same module. We can then recluster this or use agglomerative or hierarchical clustering to define different levels of hierarchy in such an arrangement. What I find very appealing about this construct here, which again we in our lab use quite routinely for assessing modularity in matrices, whether they are structural or functional, is that it allows us in one single snapshot capture the multi-scale architecture. We can recluster this as shown here on the right, which gives us different levels of scale. Depending on where we cut this tree, we can find larger or smaller modules in this particular arrangement. A general point is that when we talk about modules in the brain, there are no hard boundaries around them. They are not as well defined as they might be in a toy model. Instead, depending on what our setting of the zoom lens is, which scale we are interested in, which scale we are zooming in on, we may find modules that are more compact and many more of them, or we may find modules that are more spread out and more diffuse. And there is really no a priori argument to argue for one reality over the other. The multi-scale organization of brain networks is truly multi-scale in the sense that at any given moment, those scales coexist, in my view at least. This does not prevent us from zooming in on them, of course, analytically. And that has been done here in our ongoing series of studies with Larry in the rat brain, and it's also done in function networks, as we'll see tomorrow. In the work with Larry, one thing that pleases me immensely as someone who is more of a computational scientist, that someone like him, who is a true neuroscientist who has studied the rat brain since the 1970s, can actually take these hierarchical arrangements that I've shown you in the previous slide and look at the component brain regions that you see here with anatomical nomenclature, which is arcane to most of us. And you can look at this and say, oh yeah, that's a circuit that we know about in rat brains that has to do with pheromonal sensory motor behavior. And so even though we derive that circuit from a data-driven analysis that's blind to behavior completely, just driven by topology, by anatomy, there are connotations of these elements, of these, I'll go back one slide, of these modules that you see here. There are connotations that these modules have, associations that they have with specific behavioral domains and specific behaviors and cognitive operations. This is very pleasing to me because it speaks to the neurobiological validity and value of these types of analyses. A couple of words at the end, then I'm done. Going over a little bit here. I hoped that was going to be done by a few minutes ago. Just a couple of slides to finish up. There are many other ways to do modularity, okay? This is just one way of defining what is a community, an internally densely connected set of regions. That's not the only way to define modules. This is what we see here in this diagram, a random graph up here. Here's a adjacency matrix that has this type of module wired in, and a trivial example and the layout on the right. But we can also have modular or community arrangements that are not what is called assortative, that have core periphery structure shown on the lower left, or have even disassortative modular organization where modules are not defined by internal density, but by internal lack of density. And there's very different modeling approaches, so-called statistic block models, for example, that allow us to identify those types of architectures in a data-driven way. These are very different models from what I've talked about with modularity maximization. They really do a much larger search of the possible configuration space and often perform principled methodologies for model selection and model identification at the end. However, they are also extremely time-consuming, just computationally, and difficult to apply in any, I would say, in any really large context, or with larger samples of brain networks or other networks for that matter. They are really, we've invested some time, my grad student Josh Faskowitz and we've spent quite a bit of time on stochastic block models and come a certain way, appreciate their strengths relative to the more simple modularity maximization schemes we've discussed so far. But to be honest, we've kind of left them aside because we cannot actually apply them in any real world sense to the kind of data that we really want to work with. Good. So I've come to the end of today, and I would like to just use this last slide as a preview of what's coming next tomorrow. I've talked mostly or exclusively really today about the anatomical skeleton, in this case shown here on the left, a lateral view of the human brain and its interconnections shown as a graph. And of course, that's a fairly static arrangement. Our anatomy, the brain that you had this morning when you got up and the brain you have between your ears right now, the anatomy is pretty much the same. I mean, okay, give and take, you may have lost a few neurons or a few synapses along the way, or something may have been rewired, you formed a new memory, who knows. But by and large, at the level of this scale here, the large scale of pathways between brain regions, your brain has remained pretty static. But what your brain is doing is changing all the time, including right now. This is a movie taken of a brain of a participant in an MR scanner who was actually doing nothing in particular, just keeping their eyes open and staying awake. And you're seeing about 10, 15 minutes of that activity unfold. The yellow splotches here are points of high activity. The blue splotches are points of low activity. And over this period of time, a few minutes of time, this person's brain has, has undergone wildly different states of activation and co-activation. And it is that dynamics, that ever-changing, that ebb and flow of activation and co-activation and statistical relationships, information flow, communication patterns that will occupy us tomorrow. It unfolds on a fairly static anatomy, at least on this time scale here, fairly static anatomy, and yet it remains dynamic, flexible, task, and state dependent. And that will occupy us tomorrow. Some of the same tools will carry over, but as I've mentioned, graph theory mainly applied on the left, community detection and modularity, yes, communication models, yes, but a lot of graph theory actually is, for reasons I'll talk about tomorrow, is actually not appropriate for this data domain. So with that, I'll stop and go back to you for your questions.